All right, so everyone, thank you for attending the webinar today. If you haven't already, please visit our website, www.denovosoftware.com, and you can download a free trial of the software there. That way, anything that you see today, you can do on your own back in the lab or back in your office. And we also have many tutorials and sample data sets and resources for customers at our website that will help get you started. So today we're going to cover proliferation analysis with FCS Express version 4. We're going to try and do that in the next 30 minutes. FCS Express makes this type of analysis very easy, and that's what's going to let us teach you how to do this in about 30 minutes. So let's just give a little bit of background about proliferation analysis. The introduction of fluorescent cell staining and division tracking dyes has made it possible to monitor the number of cell divisions during proliferation and to really examine the relationship between proliferation and differentiation. Software such as FCS Express are going to help us better describe and analyze that data. And with each cell division, the whole premise behind proliferation analysis is that when you stain a cell with a proliferation dye, when that cell divides, the dye is divided roughly equally between the two daughter cells. And therefore, the dye fluorescent intensity is really a measure of the number of cell divisions that any given cell has undergone. So after staining, the cell population is analyzed, usually at regular time intervals by flow cytometry. A histogram of the number of cells as a function of the dye fluorescence intensity can be generated. That's what we're going to be plotting in FCS Express. And typically, cell proliferation dyes can be used to track up to 8 rounds of division before the fluorescent intensity is reduced to the autofluorescence of the unsane cells. FCS Express proliferation module provides curve-fitting algorithms, and this is going to help us estimate the proliferation fraction in your sample. One thing to note is that with FCS Express version 4, the proliferation module now comes free with every professional version of FCS Express. You don't have to purchase that as an extra add-on if you're getting the professional version. So keep that in mind. And I think we're just going to get started here and go right into the analysis. So what I'm going to do is first open up a new layout. And keep in mind as we're going through the analysis today, we're kind of under the assumption that you already know how to insert plots and make gates, maybe even use tokens. We'll explain some of this in detail, but for the general purpose of this webinar today, we're going to assume that you know how to do most of these things in FCS Express. So the first thing we're going to want to do is load up our proliferation data set. We have some proliferation data that is available in our tutorials and sample data set. That's what we're going to be using today. And I'm going to come into the data list here. I'm going to load up those samples. And you'll notice that we have 4 samples here. I'll get them loaded into the data list and tell you a little bit about these. So of these 4 samples, we have an Unsane Control. You can see that right here. And the Unsane Control is exactly as it sounds. It hasn't been stained with any proliferation dyes. This Unsane Control is going to provide us a background or a reference for our background kind of autofluorescent staining for the experiment. We also have a cell trace violet control. So cell trace is the proliferation dye that was used in this assay. And this particular control is on a sample where the cells have not divided. So this is going to show us kind of our starting generation or the starting peak from which proliferation is going to occur. Then we have 2, I guess we're going to call these samples today. One is a stimulated control. This stimulated control is the cells have been stimulated with a particular proliferation drug, and it's going to serve as a control that the experiment worked. And then we also have a sample in here that we're going to analyze in the end. So with these samples, we're going to create a proliferation analysis. We're going to format some of these plots, and we're also going to use tokens to assign the background and starting generations based on the controls. So based on the Unsane Control and based on the cell trace violet control. Once we do that, once we set this up, we're going to do a batch analysis of these multiple data files. We're going to put these out to PowerPoint. We're going to put statistics out to Excel. We're going to show you that we can save this analysis so that you can use it as a template in the future for other analysis. So let me erase these drawings, and we'll get started here. The first thing we're going to do is we're going to create 2 pages. One page is going to be for the Unsane Control, and that's going to allow us to come in and analyze just that data separately if we want. The next page is going to be for the stained, or the cell trace violet control. We're eventually going to come in and insert a 3rd page for analysis. So let's get right into it. Let's insert a plot. I'm going to do that by dragging and dropping from the data list here. We're going to insert a color dot plot. And you'll notice this just looks like a typical flow cytometry plot. We're going to come in here, and one of the important things for proliferation analysis is you only want to be analyzing your live cells. So we're going to set up a standard gate here, and this is going to be the side scatter versus forward scatter live cells. You can notice we created that. And I'm also going to insert a gate view here so you can see the gating hierarchy as we create this. And we also have a number of parameters that we can choose from in this data set. You can see that it was stained for CB4, the cell trace violet proliferation control, and something called SiteOx Advanced. What I'm going to do is insert another plot gated on the side scatter versus forward scatter population by dragging and dropping the gate out. And we're going to come to the SiteOx Advanced parameter. And what this SiteOx Advanced die does is it's an exclusion die. So cells that are live do not take up this die. So it's a way that we can further come in and gate and define the live population of these cells. So what I'm going to do is select this negative population, and again this is going to further refine our gating, and we're going to call these live cells. We'll mark it with SiteOx. And you'll notice in our gate view that we've created the side scatter versus forward scatter live, and then we have the live cell SiteOx stain gate underneath that. So this is a very good representation of all the live cells that we're going to want to use for our analysis. Now what we're going to do is we're also going to insert a histogram for this data. I'm going to put this right here. We're going to look at the cell trace violet, or the proliferation die, and we're also going to apply the SiteOx gate to this. And now we're really only looking at the live cells. And again, you'll notice that this is the Unsane Control. That's why we have only one peak here. This is representing the background autofluorescence for these cells in this particular experiment. And since I'm setting this up as a control page, and I may want to use the mean fluorescence of this population later on in the analysis, and I'll show you how we're going to do that. But what I want to do is create a marker on this population. I'm going to say, here's my negative control. I'm also going to come in, I right-clicked on the plot, I'm going to choose Format, and I'm going to format the marker, and instead of M1, I'm going to call this negative. And you'll notice that my marker is defined as negative here, and the statistics for that marker are going to update as I change that around. Now, we're going to also create a page for the positive control. Later on in the analysis, when we do the batch of this data, I actually don't want these data files to change. I actually want these data files to stay the same because they're going to be my negative and positive controls throughout the entire analysis that we're doing. So to do that, make sure that these stay the same during the batch process. I'm going to highlight these plots. I'm going to come to Format and Overlays. I'm going to uncheck Change Data File during Next, Previous, and Batch. That way these plots always stay the same. They're always based on the controls. So I did that for my dot plots, and I'm also going to do this for my histogram. And another nice thing I can do down here in version 4, we have these page tabs. So if I right-click and choose Format, I can call this Negative Control. That way we know what's on each of our pages. So we've set up our negative control. Once we've done this, it's very easy to actually duplicate this for the positive control. I'm going to highlight all the plots. I'm going to copy these. I just pressed Control-C. I'm going to open a new page and press Control-V, and we've pasted all those plots in the new page. Let's rename this page because this is going to be for our positive controls. And what we want to do now is change the data on all of these plots so that they reflect our positive controls. To do that, you'll notice that these are still selected. I'm going to select my positive control from the data list. I'm going to choose Change Data on Selected Plots. So I've changed the data, and you can now see there's many less cells in this positive control. Obviously, they didn't collect 50,000 cells for this analysis, but they're all appearing here. Our negative gate now doesn't apply, or a negative marker. We would have to move that up here. And we're going to narrow it in on this population because this is the undivided control. And we're also going to format by right-clicking on the plot, and we're going to change this marker to positive control. So you'll notice now that we have a positive control population defined on our positive control page, and if I do a multi-view, we can actually look at both of these side by side, and you can see that we have our positive control and our negative controls. And again, this is going to help us define populations that represent the controls for our entire experiment in our next page that we're going to do the analysis in. So let's create one more page, and I'm going to call this page Sample Analysis, and we're going to set this up to actually do all of our proliferation analysis again. So in this case, I'm just going to insert some plots real quickly. We're going to show the forward scatter versus side scatter. I'm going to right-click and choose Show Gates to show the gate that we set up. Again, I can just drag and drop that out. We'll change our parameter to the SiteOx gate. We'll right-click, and we'll show the live SiteOx cells. And again, we want to change our data real quickly because we want to start working with our control samples. So I'm going to select all the files here. I'm going to choose my stimulated control in this case and say Change Data on Selected Plots. So there we go. Our data for our sample proliferation analysis is in here. What we next want to do is insert a proliferation plot. So to do that, it's very simple. We just click the proliferation plot button up here under 1D plots in our Insert tab. And we'll draw a plot. And we're going to have to switch to our cell trace violet parameter. And you'll notice when we do that, the FCS Express has inserted some different overlays here. You can see that there's the green check pattern, there's this black overlay that's in here, there's a red line that's in there, and there's also some blue in the bottom. These are all different overlays that FCS Express is representing for proliferating fractions. I'm just going to show you what those overlays mean real quickly. To do that, I right-clicked on the plot. I'm going to choose Format. And let me bring this over, and we'll look at the overlays. So the first overlay, which is in black, you can see here that we've represented that in black. That's the actual sample. That's the actual histogram that the data represents. Our fit, or our proliferation fit, is represented in red. You can notice that that's the red line that's represented over all of the data. We're also representing background noise from this proliferation analysis in light blue, and that's what's being displayed in this overlay here. And every other generation that we've defined here is displayed in a green color, and we're using this hatch mark fill style for that. So that's all the individual generations appearing here. I'll just draw your attention to that, and you can see that here's one, here's another, and there's 10 of these in there. I'll tell you more about that in a few minutes, but you can see that that's what all the different colors for proliferation analysis are representing. One thing that you'll notice about this analysis is it doesn't look very good yet, right? And this I want to draw your attention to is because this data is still ungated. If I come up here to my gating tab, you'll see that there's no gate. And it's very, very important to gate on the live cells in a proliferation analysis. If I drag this gate the SiteOx Live gate on here and apply it, you'll notice that our proliferation analysis immediately became a lot nicer and a lot more of what you would expect out of a proliferation analysis. Another thing I want to do with this plot, you'll notice there's a lot of blank space out here in this particular plot. I might want to zoom in on this a little bit, so I'm going to right-click on the plot and choose Magnify Resolution. And we'll just grab this section, and we'll zoom in on that plot just a little bit more. This will leave us some area here because we know the autofluorescent population is down there somewhere, so we'll save that for that particular population later. Now, the next thing I want to do, I want to get some statistics from this data. Before we start making any changes to the proliferation fit or the analysis, let's see what the statistics look like. We can get statistics by right-clicking on the plot, on the proliferation plot. You'll notice that we have dedicated statistics besides the standard histogram. One is proliferation fit statistics, so I'll choose that, and we'll put this right up here. And let's move some of these plots around so we have plenty of space to work with on the page. And you'll notice here our proliferation fit statistics are telling us about the division index, proliferation index, a whole bunch of statistics. We describe what all these statistics are in the manual. I'll show you how to access that a little bit later. But if you right-click on these statistics, you'll notice that we also have the ability to choose degrees of freedom. You can check or uncheck anything from here. So if you don't want to view certain statistics, you can do that. And the one other statistic that we have that everybody's going to want is the ability to look at the proliferation population statistics. So what this is going to do, it's going to create a table. I can put this table anywhere I want, just like moving around a text box in PowerPoint. And you'll notice that it's showing us the different generations, the peak channels, values, the number of cells in each generation. I can actually right-click on this. If I want to get the number of original cells, we can get that. So the software is doing all these mathematical calculations in real time as you insert these plots. They're going to change as you change fit parameters. They're going to change as data files are changed. So that's all available to you here. Now I want to show you a little bit about the fit parameters so you can see how these things work and how, you know, if your data doesn't look like this kind of perfect case of cell proliferation, which is generally the case, you can come in and change those. So to do that, we can right-click and choose Format. And let's bring this over to the side a little bit so you can still see the statistics in the plot. And you'll notice that in the proliferation section here, we have an option for fit parameters, and we have an option for position markers. So the fit parameters are going to allow you to change or manually enter parameters associated with the data. The default for FCS Express is to calculate all of these things for you the best that it can. As I said, sometimes you're going to do proliferation experiments where you know, the software may not do the best calculation by eye, so you may want to come in here and change these things. These are all described in the manual. I'm going to give you a brief overview of what they mean. The max number of peaks, this sets a limit for the number of divisions to analyze. For this particular data set, 10 will work great. You know, if you only analyze your cells for, let's say, 3 or 4 days and there was only 3 or 4 divisions, you would come here and change that to the number that you kind of expect. It gives FCS Express a starting point to make some assumptions about your data. We also have this Use Fixed CV checkbox. So when you insert the data, the default is to calculate the CV of each individual peak or the spread of the data. If you wanted to change that, you can actually check this Use Fixed CV button and you can enter your own CV. If I click 6, you'll notice when we preview the data that the peaks actually became much more narrow because we're using a fixed CV value of 6. This can be useful in some cases. In this particular case, if I use the default, FCS Express does a very good job for that. You'll also notice that we have the ability to use same CV for all peaks. This is going to use the CV value that was calculated or manually entered for all of the peaks within the analysis. We also have the option to use a fixed peak ratio or a peak ratio that was determined by the software. In this case, the peak ratio is 0.52, and in an ideal situation, when you're looking at proliferation analysis, each peak should kind of be half of the dye intensity of the last peak. So a perfect theoretical value would be 0.5. In this particular case, it varied a little bit. It's 0.52, but if you wanted to change that, you would just click the button and maybe say 0.4. You can see that the plot is going to update, all the stats are going to update, but my value of 0.4 really doesn't make any sense here, so we're going to allow the software to calculate this for us. We can also choose to use the same peak ratio for all of the peaks. If we wanted to do that, we would just check this button. That way you would use this 0.52 value or whatever value you entered manually to calculate the peak ratios. And you'll also notice that we have the ability to use a fixed starting generation or to use a fixed value for background noise. Now you can manually enter these values in here. If I wanted to come in and enter a value, I could do that. But this doesn't really mean much unless you're going through and looking at the data and manually kind of calculating where that fixed generation starts. The better thing to do here would to actually be use your controls. The controls that we set up on the positive control page down here and the negative control. The positive is going to define the fixed starting generation, and the negative is going to define the background noise. So let's do that. And let me show you how we can use tokens to do this. So I'm going to delete this value, and I'm going to use the fixed starting generation, and I'm going to say use a token. And it's going to be a statistic token. And that token is going to be from my positive control. And this is my fixed starting generation you can see here. The statistic that I want to use is the positive control marker, and I want to use the geometric mean. And for my display text here, I actually want to use a token value because we want to use a number. And you can see that this is the number that was determined from our positive control sample on the positive control page. And we're going to do the same thing here. This time we're going to use it from our negative control plot. And we're just going to find that. We can see in the thumbnail here, here's our negative control. The statistic again is going to be the negative marker and the geometric mean, and we're going to use the token value. So when we do this, I'm going to click preview, and you're going to notice immediately that the statistics down here are going to update, that the plot is going to update, and just keep an eye on these things as I choose to preview this. And you can see that there wasn't much change to the plot, but our statistic values change. And this represents a more accurate way of doing your proliferation analysis because your starting generations and your background noise is actually generated from data within the same experiment. So this is a great way to use controls to empirically determine where these values should be coming from. And let me erase some of those drawings I made. And another thing I want to point out now is that we have this fixed starting generation and the background noise now defined in the software. And we can represent these as position markers. And that's the next tab in our categories here. So these position markers, you can have position markers for the undivided cells peak in the background. If we want to show that, we would just click Show the Position Marker on the plot. I like to make these a little bit wider so it's easier to see. When I click Preview, what you'll notice here is that our undivided cells peak is falling right over the first generation, or the undivided cells. And this means that our controls worked very well since this value is coming from our control page, and it makes sense and it aligns with the data, and we can visually represent this with this bar. And let's do that also for the background. We'll show this on the plot. In this case, let's choose a different color. That way we don't get confused. If I bring this over and we'll click Preview, you can see that this is where our background generation or our background autofluorescence number is coming from. And if we were to overlay this on the negative plot, you'll see that this will align with right in the middle of where that marker value was defined. So I'll click OK. That's going to apply all those changes. And this is a great start for analysis now. So we've covered a lot in the last 20 minutes here. I just wanted to pause. Are there any questions that anybody has for anything that you've seen so far? We've pretty much gone through how to create statistics. We've gone through what the different fit parameters mean, how to insert or use tokens to define starting and background generation. So it's a lot to take in a short amount of time. And I don't see any questions coming in, so we'll just continue on. Now, another thing we may want to do with this plot is we may want to format it so it's a little bit easier to see the different generations that we've defined. Right now, they're all represented as the same green color with the same green check marks. When you publish this data or you present it to colleagues, you may want it to look a little bit different, a little bit nicer. One thing that we can do here is if I right-click and choose Format and I choose Overlays, you notice that we have access to all the different generations and that we can color these. And this is a very typical way to show proliferation analysis, is to color these different populations as different colors. And to do that, I'm going to choose to fill the histogram area with a solid color. You notice that's the first block that we have available there. So this first generation is going to be green and solid. And I'm just going to go through here and I'm going to choose some different colors and make these solid for all the different generations. And this will only take a minute. And once you've set all these up, you'll see that when we save this layout as a template, you'll never actually have to do this again. You won't have to come in here and change the colors. This will all be saved with the layout. So once you find a color scheme that you like, you can save it, you can keep it there, and have that for posterity. But this only takes about 30 seconds, and we're just about done here. And I really like the transformation when we do this. If we click Preview, you'll notice that we colored all the different generations of the plot as different colors, and it's going to make it a lot easier to see where your different peaks are and where they fall. Now, I also want to draw your attention to something here. This is kind of it can be a pitfall for proliferation analysis. You'll notice that our histogram is kind of choppy, and that's fine. That just means that we have enough data and the resolution in these plots is good. What you may be tempted to do is actually change the smoothing option to actually smooth this histogram. And I'll do this. We'll make it 5 and click Preview. And you'll notice that our histogram was smooth and it looks a little bit nicer. But what I want to draw your attention to is the statistics for this. If you watch our statistics table down here, and we change our smoothing back to 0, our statistics are going to update, or they're going to change. That's because when you smooth the data, the proliferation fit statistics are actually based on that smooth data rather than the raw data itself. So while this may be tempting, we really want to recommend that you just use the raw data as it comes in. If you want to view a smooth histogram, maybe for a presentation or something like that, just insert either another proliferation plot to do that, or insert another histogram to do that. All right. So we've made our histogram look beautiful. We're ready to send this out to our colleagues. We're ready to send this out to the publisher at this point. So what do we want to do? We want to analyze all the rest of the data that's in this data set. At this point, we're just looking at the cell trace violet stimulated control. So if you want to go through and look at how the different data sets here or the different data files look in your analysis, you can come through and with our data tab here, we can just choose previous and next, and you can see that we're on the cell trace violet stimulated control. So if I use next, we're actually going to go to the stimulated sample. And you'll notice that our proliferation plot updated, all of our statistics updated with this new data. And if we wanted to go back through and check that this was working out with our control, you can see that our control still worked very well. We only have one population or one generation of undivided cells shown here. And again, if we go through and we look at our Unsane Control, you can see that we actually did a pretty good job of determining that as well. We can do a better job here, but I'm going to save that for the batch process to show you how we would do that. So that being said, we're going to go right through to the batch process now. So we're going to batch all 4 of these data files together. And we're going to do that through the Batch tab, Batch Actions. And what I typically like to do is I want a PowerPoint report, and I also want some of my statistics to go out to Excel at the same time. That way I have a kind of running lab notebook of everything I've done. So let's say save the PowerPoint. We'll open this after saving. And in this case, we're really only interested in page 3 for our sample analysis. Pages 1 and 2 are our controls. If you wanted to save them separately, by all means, do that. I also want to put all these in the same presentation. And I'm going to save this to my desktop as Proliferation Experiment. So that's our export to PowerPoint. That's all set up and easy. The next thing I'm going to want to do is actually send some of these items to Excel. So I'm going to choose Export to Excel, and we're going to say Proliferation Stats, and we'll click Save and click OK. And now all we have to do is just say what do we want exported to Excel. FCS Express version 4 makes this very easy. We can drag and drop a lot of things here. So if I wanted to get, say, the sample name, all I have to do is for my first column in Excel is drag that there, choose statistic token, choose file name, and we want to give this column a name, so we'll call this sample ID. And our first column in Excel is going to be for the sample ID for whatever sample happens to fall in this plot. Now I also want to put in the division index here, so all I'm going to do is click and drag that into the Excel column mode, and now our division index statistic is going out to Excel. All I have to do is give this a field name. We want it to have a nice column header when it gets in there. So we'll choose division index. If I want to do the proliferation index, you can see I can put that in there as well. We'll call this proliferation index. And that might get a little tedious doing all these one by one. So another thing you can do is hold down Control Shift as you select multiple statistics, and then just drag them into the column mode at the same time. So we just dragged in the percent divided and the peak ratio. All we have to do at this point is give that a field name. So when our Excel report is created, we know what these columns represent. And that's that. We're ready to do our batch analysis. It's going to be going out to PowerPoint and Excel with these 4 statistics. What I want to do though is I'm going to come into my batch options, and I'm going to choose the pause between iteration. Just because I looked through this data beforehand, and I know that maybe some of these gates might need to be shifted around while we run the batch process. And I'll click OK, and we can close this. And it looks like everything is set up. Let's adjust our live cells gate a little bit, and you'll notice that the plot updated. Let's run the batch process. So we've run the batch process. Excel is open on my other screen. PowerPoint has opened on my other screen. We switch to the next data sample. You notice, maybe we want to adjust this gate a little bit, include some more of those cells. That's going to update all of our statistics here. And we're going to keep this change for the next sample, and we'll click Continue. So we switch to the next sample. In this case, let's make our live cells gate a little bit more lenient, and you'll notice again all the statistics are updating. We'll keep this change, click Continue, and then we're going to come into our Unsane Control. And in this case, we're going to want to make some changes. We notice that the software may not have done the best job finding all the different peaks in here. So one thing I may want to do is actually go to a 2-page view. Maybe we have to adjust our negative and positive controls. Since we set this up, if you remember, the negative background population and the positive population are based on tokens for these histogram markers. If we adjust this, it's actually going to change our data in real time, right? So we can maybe make that a little bit tighter to narrow in that population. Maybe we'll come in here and make our positive control a little bit tighter as well, and that didn't make too much of a difference. But what we can also do is, since we're in the pause mode for the batch here, we can also format this, and let's take a look at our fit parameters. In this case, let's just uncheck everything and click Preview. And when we do that, when we unchecked everything, we kind of put this manually and we said that this is the background population, we get a very good representation of the data now. It makes sense. There's no proliferation statistics available here because there are no dividing generations. This is all the background population. It's all centered along this center line. And there's no proliferation statistics here because there's nothing proliferating. So this data makes a lot more sense. And since we pause this, but we're still in the batch mode, we can click Continue. And that's going to finish our analysis for all 4 samples. PowerPoint's going to open up. You can see that happening here. We now have our 4 different samples that we analyzed. If I click through PowerPoint, you can see that all the statistics were put out for these things, and we have a nice little report. And let me bring over from my other screen that we have the Excel table, and if I can just grab this, you can see that our sample ID went out. We have our division index, proliferation index, percent divided, and peak ratio. And this all makes sense, right? Here's our control. Here's our proliferation index. We only had the starting generation. And our Unsane Control, there was nothing going on because it's our Unsane Control. And here's our 2 other populations, or our 2 other samples that we put together, and the statistics went out in real time. So this is how you can quickly and easily generate plots or reports and statistical analysis for data very quickly. And at this point, if we wanted to save this layout, we can save this as, we'll just save the layout. And I think that opened up on my other screen here, and we'll call this sample analysis. Once we've done that, this is not just a layout for this particular data set of 4 samples that we used. This can also be used as a template. So if next week we ran a similar experiment, we're going to have our good negative controls and positive controls. We would just come in here and select all of these, change the data on these files as I showed you earlier to be our new Unsane Control. Select these, change the data to be our new positive control. And for our sample analysis, I mean, if we wanted to come in and say insert data from a new experiment, we'd say, let's use the sample analysis from this date. We would select these plots, choose that data file, and that's going to update everything. And now we're off and running with our experiment for our new data set. In this case, if you remember, this probably doesn't look so good because we changed our fit parameters to use some different values, and that should look a little bit better. So that pretty much covers most of the proliferation analysis in FCS Express. If you remember, what we did here was we inserted plots based on for the negative and positive control populations. We use those plots to help define where those generations are in our sample data. We formatted these plots so they look very nice. We have everything on one page, and we actually exported all that data to Excel and PowerPoint just at a click of the button. And using the pause feature in the batch analysis, we're able to make changes on the fly rather than having to analyze the data and then go back and make those changes. So are there any questions at this point on anything that we went over today? If not, I'm going to point you to some further resources that we do have for proliferation analysis. So all the data that you saw today can be found in our tutorials. They can be accessed through our applications button, help, tutorials, or on our website. We have on our Novo software website here, under support, you can access tutorials. This is going to bring you to the same spot. But what I want to draw your attention to is that here's the proliferation analysis tutorial. This is going to describe the experiment that we did today, or the sample data that we used. It's going to tell you all about that if you have more questions about what was actually done with that. We're going to show you how to insert proliferation plots, fitting cells, formatting proliferation plots, and even doing the batch process at the end. We also have, you'll notice that this is our tutorials and our manuals, that we have a proliferation analysis section. This is going to tell you a little bit about proliferation analysis. It's going to tell you about the add-on, which again is free in the professional version. We're also going to describe the fit parameter options in more detail. And we're also going to describe in great detail the proliferation statistics that were used to generate all of these plots. So if your publisher or your colleagues come back and they said, what happened mathematically in the background to generate all these things, you can refer them to this page. You can use this page to publish in your publication, whatever you want to do. But all the details of everything that happened in the analysis are available in the manual. Now, besides the manual, there's always us. We like to pride ourselves on our customer support. If you have any particular questions that weren't answered in the webinar today, or just simple questions, how do I insert that starting generation token or something, just contact us at support@denovosoftware.com and we'll typically get back to you in 24 hours. And again, you can always download a free trial, or your colleagues can download a free trial. If you like what you saw today, you can tell them. You can get that from www.denovosoftware.com under our download tab. And again, the professional version and our trial version are going to have the proliferation module for you in there. So at this point, are there any other questions that we can address here? It looks like we may have had one or two here that were answered. And if there aren't any more questions, I think we're going to close the webinar today. And again, if anything comes up after the fact that you think of, please email us at support@denovosoftware.com. We're more than happy to answer all of your questions. So thanks everybody for coming out today. I hope you were all able to take something away from this webinar and that all of your proliferation analysis will go quickly and smoothly in the future. I know how difficult it can be to do that in the lab, but FCS Express will make the analysis side of things very easy.